welcome to the deep dive. Okay, imagine for a second. Think about your brain having its own, really sophisticated security system. A kind of internal border control, right? Deciding what gets in, what stays out. It's pretty amazing biology.
It really is an incredible feat.
But what happens when that system, this, protective barrier, what if it starts to go wrong?
That's a huge question because, when that shield gets compromised, the consequences for the brain, they can be really profound. It flips from guardian to, part of the problem itself.
Exactly. And that's really the core of our deep dive today. We're looking into Parkinson's disease and specifically focusing on this amazing brain barrier and some pretty groundbreaking new research. It seems inflammation might be a key reason it breaks down.
Yeah, our mission here is to unpack some recent findings. There's a key study, Lifespan.io, highlighted, to really get a handle on how scientists are trying to, seal the barrier, potentially.
Right, to break that damaging cycle in the brain.
Exactly, disrupt that vicious cycle.
So if you're after a quick but still thorough look at some complex, cutting-edge science that could genuinely change how we think about neurological conditions, you're definitely in the right place.
Yeah, let's dive in. It's fascinating how the brain's own defense can become, a vulnerability.
It really is. Okay, so let's start with the basics. This border control, the blood-brain barrier, or BBB. In a healthy brain, how does it work? What makes it such a good guard?
Okay, think of the BBB as the brain's super exclusive security checkpoint. It's this incredibly tight network of cells lining the brain's blood vessels.
Okay.
And its whole job is to strictly regulate what gets from the blood into the delicate brain tissue itself. So normally only very specific things get through: nutrients, oxygen, certain molecules.
Stuff the brain needs.
Right. It's incredibly selective, designed to keep out pathogens, toxins, lots of large molecules. Basically the riffraff.
That sounds vital. But you mentioned in conditions like Parkinson's, it can become dysfunctional, leaky. What actually happens when the security system springs a leak?
Yeah, leaky is a good way to put it. When the barrier gets compromised in Parkinson's, it's a major issue. Things that are normally kept out start slipping through.
Like what?
Well, harmful compounds, maybe misfolded proteins that shouldn't be there, certain immune cells, inflammatory signals.
Wow.
And once these things cross that weakened barrier, they just add fuel to the fire. They contribute to more damage, especially to those dopamine-producing neurons that are so central to Parkinson's.
So it's not just a side effect. It actually makes the disease worse.
Absolutely. It becomes an active accelerant. It drives the progression.
That leakiness, wow, it really kicks things into a higher gear.
Right.
Which I guess brings us to this dangerous feedback loop you mentioned, this vicious cycle of inflammation and barrier breakdown.
Exactly. It's a nasty cycle.
Can you walk us through how that works? How do these things conspire against the brain?
Sure. So a key player in this whole destructive process is a molecule called TNF-alpha.
TNF-alpha.
It's a pro-inflammatory signaling molecule. Think of it like an emergency flare that the immune system fires off when it detects damage or infection.
Useful sometimes, then.
In the short term, yes. But the problem is persistently high levels of TNF-alpha. They're linked to chronic inflammation and, unfortunately, in neurodegenerative diseases, to more neuron death.
So it's like an alarm bell that just won't switch off. How does that create this self-feeding loop?
Right. So it starts like this. In Parkinson's, as those dopamine neurons start to die, they release distress signals.
Okay.
The brain's immune system responds. But sometimes it overreacts. This leads to a surge in inflammation, and TNF-alpha is a big driver of that.
And the inflammation itself causes problems.
Yes, because that increased inflammation then makes the blood-brain barrier even more permeable, leakier.
Okay.
And once the barrier is weaker, guess what? More immune cells, more inflammatory stuff gets into the brain.
Which causes more damage and more inflammation.
Exactly. It fuels this deeper, really destructive cycle. It's a terrible loop for the neurons caught in the middle.
Wow, that sounds like a perfect storm inside the brain.
Yeah.
Okay, so if inflammation, and specifically this TNF-alpha, are so central, have researchers tried targeting them? What about, TNF blockers?
Yeah, that's the logical next step, isn't it? And yes, they have definitely looked into this.
They have.
And in mouse models of Parkinson's, studies have shown some promising results. Blocking TNF-alpha actually seemed to help preserve the blood-brain barrier's integrity.
Really?
Yeah, and it reduced brain inflammation significantly in those models. There's a well-known TNF blocking drug called etanercept. It's already used for things like arthritis.
Etanercept. Okay. So a drug that's already out there seemed to work in the mice, protecting neurons, reducing inflammation. combination, that sounds huge.
It does sound hopeful, absolutely.
My sense of butt coming. As always with mouse studies right now. What are the big hurdles here? Why can't we just jump straight to using this in people?
That's the million-dollar question, isn't it? And yeah, there's a major limitation. It boils down to the barrier itself, the blood-brain barrier in mice. It's just naturally leakier, more permissive than the human one, especially in the specific brain regions that are hit hard by Parkinson's. So drugs, even big ones like etanercept, might get into the mouse brain much more easily than they'd ever get into a human brain.
Okay, so access is easier in mice. Is that the only issue?
Well, no. There's also the fact that these promising mouse studies, they likely weren't using just the standard off-the-shelf etanercept. They often used specially engineered forms of the drug, modified versions.
Okay, so it's not as simple as just repurposing the arthritis drug. You're saying the standard version, the one doctors prescribe now, probably wouldn't cross the human BBB well enough.
Precisely. Standard etanercept is a large molecule. It just doesn't cross the human blood-brain barrier easily under normal circumstances. It might get through a bit if the barrier is already severely damaged, but probably not enough to have the desired therapeutic effect seen in mice. And this difference between the standard drug and these potential modified brain-penetrating versions, it's absolutely critical. It's a big reason why promising animal results often don't translate directly to human treatments. It's a huge challenge in neurology drug development.
Okay, that makes sense. So if the standard drugs are basically stopped at the gate, what's next? What are the clever strategies researchers are working on now to actually get treatments into the brain? These brain-penetrating treatments.
Right. This is where the real ingenuity comes in. Scientists are actively designing completely new ways to deliver similar benefits like blocking TNF or reducing inflammation, but getting them directly into the brain.
How?
Well, one really cool approach is called a Trojan horse strategy.
Like the Greeks.
Exactly. They essentially attach the drug molecule to another molecule that the brain's barrier does recognize and lets in. It's like sneaking the drug past the guards by disguising it.
Clever. Using the brain's own transport system against its defenses.
And I've also seen talk about nanoparticle delivery systems. What about those?
Yeah, nanoparticles are another big area. These are like tiny engineered packages, like microscopic delivery trucks.
Kind of, yeah. They can encapsulate the anti-inflammatory compounds, carry them across the barrier more effectively, and then release the treatment right where it's needed.
That sounds very futuristic.
It is pretty cutting-edge stuff. This whole field of direct brain delivery has really taken off in the last, say, 5 to 10 years.
So it's exciting, but probably not something we'll see in the clinic next year.
No, definitely not. While it holds immense promise, it's important to be realistic. These therapies are still years away from human trials. It takes a long time to ensure they're safe and effective.
Okay.
And another crucial point, these are being developed to complement current Parkinson's treatments, like L-DOPA, not replace them.
So adding another tool to the toolbox.
Exactly. The aim is more towards slowing down the actual progression of the disease, tackling that inflammation cycle, rather than just managing the symptoms like L-DOPA does.
That's a really important distinction. Wow, what an incredible journey, looking at the brain's own defenses and how they can go wrong.
It really is complex.
So just to quickly recap for everyone listening, we talk about the blood-brain barrier, its vital gatekeeper role.
The security system.
How inflammation, especially driven by things like TNF-alpha, seems to make it leaky in Parkinson's, creating that damaging cycle.
Right, that vicious loop.
And then we looked at the really innovative research, trying to find ways around the barrier to deliver treatments directly into the brain, even though translating those mouse studies is definitely tricky.
Absolutely. And maybe a final thought to leave people with. Just consider the incredible ingenuity it takes to design a drug that can essentially outsmart the body's own highly evolved defenses, like the blood-brain barrier.
Yeah, it's mind-boggling.
And this whole frontier of drug delivery, learning how to navigate or even leverage the brain's security system, it might not just be for Parkinson's. Think about Alzheimer's, brain tumors like glioblastoma, other neurological conditions.
Right, the potential impact is huge.
It really is. Mastering brain delivery could unlock treatments for so many difficult conditions. It's definitely a field brimming with potential and certainly one to keep a close eye on.